Yeah, so the idea of this group sort of started from a Canadian group of interest led by Claire Brown and Natalie Wood. And the time zone for their meetings is really bad for us. So they asked if someone would be interested in hosting a meeting in our side of the world. And that's why we started this, to see first how much interest there is, and also to share resources. So they started this Google Doc. One thing I wasn't sure if the people that just joined would be able to see the Google Doc. I can put it again. Just so you can put the attendance and you can have a bit of a look of what they've been doing in their Google Doc. The idea is that the meetings will be sort of alternated. They do a meeting 1 month and then do a meeting 2 months after if there's enough interest. In the Google Doc, apart from the attendance, there's also links to a master document where there's notes from the meetings, and you can submit forms, and you can submit fluorescent dye suggestions. Actually, Natalie just joined, who is the person that is organizing the Canadian meeting. So she's the person that started a lot of these documents as well. You had some questions, Pranali, about the document? Oh, I think I'll just mute.
Yeah, sorry. Yes, I just wanted to know, what do we need to input some information in the Google Doc?
Yes, so in the first one, the main thing would be so we have an idea of the attendance. So if you could put your name on it. And then you can also have a look through the notes from the Canadian group.
Okay.
And I'll show you, maybe I'll share my screen so you can see. So within the document, in the first page, you can see here we would have the attendance. Then if you click on that one, you can access the master document, where the idea is to share resources. Some of these ones are Google Forms, so people can say or use certain fluorescent dye or suggest speakers or topic suggestions. So then we can mold the meetings to suit more people.
Okay, I think for me, it's directly taking me to the master doc. Can't enter my name as attendee.
This one?
We only see one Google link, that's why.
Okay.
Yes, that's better. Yeah. The thing is that it's a document within a document. Probably not can correct me if I'm wrong.
Yep. What was the question? Sorry.
Yeah, so the document is sort of nested within the other document, like the master. Yeah. So you can only see one.
Yeah, one of them is for mainly the notes, and then the master Google Doc is where you can kind of access all of the forms and eventually the spreadsheets of people who have filled in the form. And so I guess, hi everyone. I'm sorry, I just jumped in there. My name is Natalie. I don't know if you have all met me yet, but I host the Canadian hosted expansion user group. So yeah, I started, so I'm a first year master's student working on developing expansion microscopy protocols, and I specifically work with brain and brain cancer tissue in mice. So that's kind of just my background, but we want to incorporate, so I actually work with the facility at McGill, which is why I've been kind of put on this project to help develop expansion and yeah, see where it goes. So nice to meet you all. I'm currently, I don't know if you can tell in the background, but it's like 10 p.m. here and I'm in a car. I'm on a volunteering shift. But yeah, I just wanted to drop by and say hi to you all. And if you feel free to email me if you have any questions, or if you want to attend the Canadian hosted times, you're very welcome to do so.
Thanks, Natalie.
Thank you, everybody.
It would also be good if people are happy to unmute themselves, introduce themselves, and just let us know if they have any experience or what are their expectations, or what do they want to learn from this group would be great. So feel free to unmute yourself for this part.
I'll start since I have video on already. I'm Gabriella's colleague at BOP at the University of Melbourne, and expansion microscopy is something I've looked into in the past, and I've never really got around to getting it started. But finding out that there's more people interested in doing it has kind of spurred me on to be more interested. Natalie, you're nodding your head, so I'm sure that's a comment you've heard a few times before. And one of the microscopes I look after in BOMP is a STORM system. And we have a lot of people looking for super resolution, but the sample preparation and the imaging setup with that isn't always amenable to every sample. So it's always good to have an alternative, and I see expansion microscopy is a very powerful and potentially accessible technique for people to use, because not everyone can get trained up on STORM. So that's my interest in it.
Thanks, Shane.
Hello, I'll go next if that's okay.
Yeah.
Hi, I'm Pranali, and I also work at the University of Melbourne. Little bit overlap with Gabriella as well. I have never done expansion microscopy, but I'm very keen to get it going. So I work with microalgae bacteria interaction, and microalgae is a very autofluorescent cellular landscape. So what I want to do is to be able to mechanically expand microalgae cells, which is typically 8 to 10 micron, and then map where the bacteria within microalgae is present. That's why I would like to try expansion microscopy.
Hey, can I go next? Hi, my name is Alina, and I'm not from Australia. I just got a link from my colleague. I'm from Israel, so it's 5 a.m. here.
Oh, wow.
But I was really keen to join this meeting because I'm really excited. I've been using expansion microscopy for like a couple of years already. I work on Drosophila, and I had success in expanding, like using different protocols and doing like 16-fold expansion also. So, and using different dyes and stuff. So I would be happy to share with you, and also to hear what you've been up to, because I'm really here in Israel, not many people experience this kind of microscopy, and I just want to hear from my fellow colleagues who use it all over the world. So yeah.
Oh, thanks for joining at this time.
Because it's just like a first group first meeting of this kind that I've ever heard of. So I was really excited. So thank you.
Hello, my name is Pam Young. I'm from the University of Sydney. I'm the light microscopy manager here for Sydney Microscopy and Microanalysis. I myself have not done any expansion microscopy, and I got very late to this meeting because I didn't realize how involved the enrollment process would be. It wouldn't just be a Zoom link. But I'm very keen. I work with about 150 some odd different researchers. So it would definitely be a good skill to have in my tool belt.
I'll go next. It's kind of safer to go after Pam because I'm also currently a manager at BOMP, a similar core facility within the University of Melbourne. Also, I haven't done any expansion microscopy by myself, and I guess I'm here today more as a moral support for Gabby and the others. But also, I think this user group meeting is really good initiative for gathering all the interest. So hopefully this group can expand further and further in the future, but I'll try to make it every time. Yeah, so thank you. Nice to see everyone.
Thanks, Ellie. Anyone else happy to share?
Hi, I'm Kalyan. I work with Gabriela as well.
I think you and Luis try to talk at the same time.
That's fine. Go ahead.
Luis, you can go first. I'll talk later.
You carry on. I'll join after you.
Yeah, so I work with Gabriela as well. We have not done much expansion microscopy yet, so we were both kind of keen on that. So Gabby is in part of that expansion group. So we want to see where it takes us.
Okay, so I'm from the University of Technology, Sydney, so director of the microbial imaging facility, but we do image larger cells as well. And Veronica Vilova, I'll save you. You can. She works with me. She set up a tissue clearing pipeline, which is the modified iDisco. She's again interested in brain tissue. So, but we've got no experience in expansion microscopy. We have an NStorm and a 3D SIM, the OMX. So really interested to get some protocols up and running.
Thanks, Liz. Maybe I haven't introduced myself actually. I started talking. Yeah, so I work as a specialist in the University of Melbourne. I have great interest in expansion, but I've really only done it in a workshop once, which was like a very short period. So it would be good for us to establish a technique, as you probably heard from my colleagues as well. The second thing I had as a plan was to share one of the presentations that was recorded from the Canadian group. So if no one else would like to share anything, anyone else? Otherwise, I'll just put on the recording so then we can watch that. It's pretty interesting presentation because it goes through a lot of the habits of expansion and things that you should be careful with. So let's see if I manage to do it properly. The other thing I wanted to let you know, so all of these meetings, they are being recorded and they've been put in a YouTube channel by the RMS. So they will be available, so you can also watch not just the recording from this meeting, but also from the previous meetings and the other speakers that have participated. The question about the mailing list from Ellie. So if you have registered on the RMS page, then you will start receiving the mails about the expansion microscopy groups.
Okay, so I'm already in.
Yes.
Just an added factor. I think we're going to be setting out kind of monthly newsletters since our meetings are going to be monthly from now on, coordinating either. So it'll be Australia hosted and then Canadian hosted, Australia hosted, Canadian hosted. So to kind of just keep track of all that and make sure everybody across the world is up to date on everything that we're offering and all of these spreadsheets, Google Forms and stuff, we're going to be sending out these monthly newsletters to make sure everyone knows where everyone else is at. And then we're also thinking of doing like a monthly feature. So if any of you do choose to, and we're also always open for people to, if they want to speak, any master student like me is obviously really encouraged to come speak about their experience or present any work that they've done with expansion. So yeah, masters, PhD, postdocs, anyone is welcome to apply to speak for us. So yep.
Yeah, thanks.
In Italian Institute of Technology in Genova.
Sure.
And
Are you guys able to hear that?
Under the supervision of Professor Alberto Diaspro and Professor Paolo Bianchini. My background is in chemistry, and now I'm doing my PhD in biophysics. And I started working last year with expansion microscopy. So this is my background. Okay. Okay, should I start sharing my screen? Okay.
Thank you.
Okay, can you see my screen?
I think we see your split screen. Okay, yeah, that's good.
Okay.
Perfect.
Okay. Okay. Okay, so today we're gonna talk about the problem of expansion validation, and especially how this problem has been faced in our group using the nuclear core complex as an intrinsic reporter. So, as we all know, even though expansion microscopy is quite recent technique, the panorama is already quite vast, and we have a lot of protocols already available. But all of these protocols start from the same point, which is the synthesis of a soluble polymer network inside your biological sample. Then can then be physically expanded, and this results in a physical magnification of the sample. So we all know that, but when you work with expansion microscopy, it's very important to see each step, to see this system as an engineerable system, and you can customize each step in order to achieve what you want to achieve. So starting from the labeling, we know that in the original expansion, the first protocol from 2015, they were using a three-functional fluorescent label featuring oligonucleotides sequence to target the primary antibodies for the immunostaining in the sample. Then, starting from that, the protein retention expansion microscopy protocol was developed, and so this allowed for the use of standard conventional antibodies and introduced for the first time the concept of post-expansion staining. And then after that, we also know that a lot of labs around the world are now developing gel anchorable small molecular probes for various biological targets. Then, speaking about the second step, which is the gelation, where you synthesize your hydrogel-based polymer, we know that most of the polymers are based on these two monomers, sodium acrylate and acrylamide, as you can see here, and then usually there is a cross-linker to regulate the expansion. But now we also know that modifying the composition of the gel and the relative amount of these monomers can have an effect on the preservation of different biological structures. Then we have the digestion step that originally started as an enzymatic digestion using a proteinase, but then in different protocols during this development, we discovered that modifying this digestion buffer, you can target it to your specific biological sample. And for example, you can supplement it with specifically enzymes to digest your sample. But in some cases, you can also completely replace this enzymatic digestion with the denaturation step, which can be either thermical or chemical. And then, of course, we have the expansion by water dialysis, and then we can finally image our sample, obtaining super resolution imaging thanks to the physical magnification of our sample using a diffraction limited microscope. So we're starting from our cell, for example, from our biological sample. In our case, let's consider a cell. And we have to always keep in mind that there is a heavy sample processing involved. And although for some application, like for example, the mapping of some neuronal network, the quantitative retention of the ultrastructure is not really needed, if we are talking about cell biology, the necessity for the validation of the ultrastructure retention becomes very important. And also you may need to be able to quantify your expansion factor in a more accurate way than simply measuring your gel size at the end of your expansion. And so in the last 2 years, a few examples on how you can validate your expansion protocol based on reference structure came out. Let's step briefly back and see how the validation was performed in the original protocol. As we know, the first validation was a comparative analysis between pre-expansion imaging taken using super resolution microscopy and post-expansion confocal images. In the first paper of Professor Boyd, and they used structural illumination microscopy as a super resolution microscopy technique, and post-expansion they used the spinning disc confocal. They use as reference structure microtubules and cluttering coated pits, and they perform a measurement error analysis, which gave a root mean square in the order of 1%, which is well within the point for expansion of the super resolution microscopic technique that they used. So we can always perform this comparative analysis to validate our expansion protocol. So for example, we can take a large image pre-expansion, and then post-expansion we can go find back the same cells, and then we can obtain something that looks like this, which is always valid. But so you first need to do a big mapping on your sample, and then you need to find back the same cells, which is not always simple. So this is a very time-consuming method. That's why we can try to use some intrinsic reporter as a method of validation. So to be able to use a biological structure as a reference, we need to know this structure very well. So in our lab, we started using as a reference structure the nuclear pore complex. The nuclear pore complex is responsible for the transport of material between the nucleus and the cytoplasm, and most importantly, for what we're interested in, is a very well characterized and known structure. We know that it has an octagonal symmetry around the axis of transport, and that it is pseudoplanar through the nuclear envelopment. Then we know the diameter of the structure, and several of the nucleoporins that compose this structure have been characterized in super resolution microscopy, and the primary antibodies to target these nucleoporins are commercially available. So in our lab, we decided to use a reference structure, the nucleoporin 153, using a primary antibody produced by APCAM. So to validate for the first time this validation method, what was done at the beginning was to simply perform a confocal imaging before and after the expansion on the same cells. Then this image can be overlapped and rescaled to a fine registration in order to perform the same comparative analysis that was performed in the past to validate expansion microscopy. Then, using the pixel size ratio between the images, we are able to measure the expansion factor on a nano, micro, and macro scale level. But what we want to do is to perform a statistical analysis in order to remove the need to do this comparative analysis before and after the expansion. So what we did was to analyze the nuclear pore complex at different expansion factor and with different optical resolution. So we performed an expansion STED experiment, and this needed, there were a lot of parameters that needed to be finally tuned experimentally. And this analysis was performed at 3 different expansion levels. Before the expansion, so on our biological sample as it was, post-digestion, since after the digestion you usually achieve a first expansion with, which in our case has an expansion factor around 2, and then at the end of the full expansion in water, which in our case has a macro expansion factor around 5. And here you can see the line profiles of these selected nuclear pore complex units compared at the 2 different optical resolution and in the 3 different expansion levels. The next step was to start this determination of the expansion factor at the nanoscale in a statistical way. And this was done using image analysis with a custom MATLAB algorithm. So what was done was to first measure the radial intensity profile of a single nuclear pore complex unit along different orientation. And in doing this analysis, the algorithm is able to calculate the radius of the pore and the position of the center. So we can have averaging all of these profile along different orientation, the final radius for each pore. Then this statistical analysis was performed on a large number of nuclear pore units, and then we can plot the sum, the distribution of all of these collected radii and fit it with the Gaussian distribution for the 3 different expansion levels. And then we can obtain the nanoscale expansion factor that we can compare with the macro scale one that we had by measuring our gel. Then we wanted to determine if the expansion was isotropy or wasn't. And this was done again using image analysis. So for each pore, we know that the maximum value of the radial intensity can be plotted as a function of the orientation. So what we did was to fit with a multi-Gaussian peak this function, and we used the first subunit as a reference, giving it so attributing the angle 0. So based on the statistical nature of the immunostaining protocol, we know that in most of the pore we will not have all of the different subunits immunolabeled. So this analysis was performed again on a large number of nuclear pore complex units, obtaining then all of the subunits in the cumulated data. So we can plot the final accumulation as a histogram, and then we can retrieve the Anglius values, which we can fit with a 7-tip Gaussian. And This is used to demonstrate the isotropy of the functions, since the angular difference among these units is a multiple of 45 of the angle of 45 degrees. The last thing we did was to try to estimate the resolution of our system. So first, we wanted to determine the optical resolution of solely the STED imaging. So to do this, in order to do this, we perform STED images on a non-expanded sample using the same imaging setting that were used for the 3 experiments: pre-expansion, post-digestion, and post-expansion. So then from the depletion curve, we were able to extract the K value in order to calculate the improvement in resolution, considering a confocal resolution of 250 nanometers. And then to calculate the effective resolution of our STED expansion experiment, we normalized the single optical resolution for the 3 conditions by the expansion factor for each condition, achieving the effective resolution that you can see here in this slide. A similar work has been done in the lab of Professor Paul Guichard using centriole as a reference structure. So in this case, the comparative analysis was based on the 9-fold symmetry of isolated center, and they used to perform imaging, direct stochastic optical reconstruction microscopy, and SIM microscopy. This a very interesting point of this work is that it shows how slightly changing the gel composition and the fixation method can have results on the different preservation of the biological ultrastructure. Because in this work, they compare the original expansion microscopy protocol with the magnified analysis of protein 1 and the ultra expansion microscopy method that they develop specifically to better preserve the ultrastructure of centrioles. Another way to quantify the expansion factor at the microscale level and to determine if there is a distortion during the expansion has been using this intrinsic in this very interesting work using fluorescent photobleaching. So what they did was to transfect HeLa cells with a plasmid to induce the expression of nuclear enhancement green fluorescent protein, and then they used 2-photon excitation to induce photobleaching of the EAGSP, introducing well-defined 3D features in the cells. So they basically bleached out some cubes inside the nucleus of their cell, and then they used these cubes as an intrinsic ruler to validate the expansion factor and the sample distortion in their samples after the expansion, measuring the dimension of this cube after the expansion. And what they found is that in their experiment, the expansion was isotropic in all 3 dimensions. Then, before introducing the last expansion factor validation method that I would like to try, I will briefly tell you what I work on now and during my PhD. So now I'm working on the investigation of modification in chromatin compaction related to genetic diseases, and I use expansion microscopy combined with other super resolution microscopy methods. Particularly, I'm working on Hutchinson-Gilford progeria syndrome, which is a genetic disease which causes the early onset of aging symptoms. And this disease is caused by a single nucleotide mutation in the lamin A gene that causes the production of the modified version of the lamin A protein called delta L50 or progerin. The presence of this protein modifies the dynamic and the structural protein, the structural properties of the nuclear lamina, especially leading to nuclear complex clustering, the thickening of nuclear lamina, and the loss of peripheral heterochromatin. Our cellular model is based on human epithelial kidney cells, and we have 2 groups of cells in order to perform a comparative analysis. In the first group, we induce the expression of the modified protein delta L50, while in the second one, we examine the wild type form of the laminase. Both are tagged with this C3-GFP, which is a modified version of the GFP protein that has been shown in the past to be efficient for cell microscopy. And to counterstain the DNA, we are using the SPI 650 DNA dye, which is a far-red minor groove binding DNA binding dye that gives very good results with the post-expansion staining as well. So it's a very interesting dye for expansion. So what we have seen is that in the HDPS sample set, there is a large variation in chromatin compaction, and this seems to affect the physical association between lamin and heterochromatin in the nuclear periphery. And seems to lead to the proper spatial separation. So we wanted to quantify this distance between these 2 structures, and we are doing this by analyzing the intensity profiles of these two structures to determine this distance between the DNA and the lamin. So our preliminary results show that the two groups seem to belong to 2 different populations. But since we are using expansion, we would like to further investigate if since the 2 structures that we are examining, and one is based on DNA and the other is a protein, we would like to investigate the different behavior of these 2 structures during the expansion. So we would like to try a microscale expansion factor validation based on DNA. So we found this very interesting work that is being carried on in the Gatta Quant Lab, where they use the DNA nanorulers, which are the DNA origami-based nanorulers that have 2 fluorophore at a very well-known distance. They are usually used to quantify the resolution of an imaging system. Basically, they use the modified version of the nanorulers covalently bonded with an acridite molecule. So these nanorulers can take part in the polymerization process during the gelling, and then after the denaturation step can be expanded. So what they did in this work is that first they quantify the macroscopic expansion factor, as we all do, and then they perform a distance analysis on a very large number of DNA origami, obtaining the macroscopic expansion factor. The interesting part of their work is that they found that there is a deviation between the microscopic expansion factor and the macroscopic one. The microscopic seems to be slightly smaller. So what we would like to do is to implement these nanorural tools inside our biological sample to be able to perform 3D studies and quantify the expansion of DNA inside our sample. So, in conclusion, we can say that in the last few years, the intrinsic reporter has been successfully used for rapid expansion microscopy validation. Although this is a process that needs careful optimization, especially if you use less robust biological structure. But you always have to keep in mind that if you have deformations on your sample, the tuning of the gel receipt you're using can mitigate this effect. And also, you can always have the possibility to add external reporters to your gel. But in conclusion, we can say that it doesn't exist a universal method for retaining all of the ultrastructures in your sample yet. So there is still a lot of work to be done. I would like to thank all of the people I work with every day, and I would like to thank you for your attention. And if you have any questions, I'm open for discussion.
I think Mattia has a question. You can unmute yourself.
Hi, thanks for your presentation. So I have like a biological question about the experiment that you're trying to do with the DNA. So
Okay.
why didn't you use a heterochromatic mark in your experiment? So why you didn't use maybe a mark for, I don't know, like lysine modification that you can tag with and you just get the heterochromatin?
I actually did that. Like here, I just show you the one with the full DNA. There is not I performed the image analysis only on this one for now, but I also stained the methylation modification on the lysine 9 of the histone 3. So well, I didn't want to talk about my work too much because this wasn't the point of this meeting, but we would like also to study the modification in both heterochromatin and euchromatin in this biological sample that we are using now.
Okay, thanks.
You're welcome.
Go ahead, Natalie.
No, I was just going to read out what people mentioned in the chat. Javier said, excellent presentation and results. Congratulations. He wanted to thank you, but he had to leave because his teaching duties called him back. But so that was the first comment. And then Chris Chan asked, how vary every time do you make like an expansion gel or the range?
Oh, I assume you mean like the expansion range, like if the expansion is the same. Is that the question? Okay, the expansion factor. Now I am using mostly the protein retention expansion microscopy protocol. So using the same condition, and I use the same also the same monomer solution because I made an amount and then I freeze them in aliquots. The expansion is usually around the same value. So every time I measure the gel, usually the expansion factor I have now is usually around 4, and usually is quite consistent. You're welcome.
Claire, did you want to go ahead?
Yeah, I had a question about the DNA origami. Just how easy is it to get it evenly distributed throughout the gel?
Okay, so this is something that we are just starting to do. We bought the DNA nanorulers with this acridite modification already, and we didn't start yet to add it into our sample. So this is something we want to do, like we are ready to do now. But in this work, they seem to be very well distributed because they put it on the cover slip, so and then they synthesize the gel, and I think we will do the same thing because we have the cover slip with ourselves. So then we will infuse with the DNA nanoruler solution, and then we will add the monomer solution. So that's it.
Keep me posted. Thank you.
George, do you want to go ahead?
Yes. Excellent presentation. That was a very nice summary of what's been done so far and a great introduction to the topic as well. I had kind of 2 different questions. One, for the nuclear pore complex labeling, are you using a primary antibody and secondary antibody complex?
Yes, at the moment, yes.
Have you considered using a conjugated primary or a nanobody if that existed, or doing post-gel staining, which has started to become popular in the literature?
Yes, we would like to do the post-expansion one, the post-expansion staining. We would like to try. I didn't try with the nuclear pore complex yet. And also we have the nanobodies for the secondary antibodies, and this is something also we're starting to use.
Okay, great. My second question is for protein and nucleic acid anchoring into the gel. I know the early protocols had kind of very specialized anchor preparation rather than the original Acra Oil X. But I know there's been 2 recent papers that have shown alternative anchors that work with both protein and nucleic acid intrinsically. Are you doing the kind of 2 anchoring step, or do you have a combination anchor?
At the moment, we are using the ACX anchoring, so it's the one based on the succinimide ester. And we didn't try. Well, we tried the first one, then NAHS. But now we move to the second one, which is the one that they use in the protein retention expansion microscopy, which seems to be better the results for us. But we will also try to do something else. I found an interesting work they used to combine expansion microscopy with Raman, so this is a different kind of work. But they seem to have a very high protein retention there, so this is something I would like to try. I don't know if I answered your question.
Yeah, that's very helpful. Yep, thank you very much.
All right, so Michael Cameron actually had a question, but for some reason his hand raising option wasn't available. So Michael, do you want to proceed?
Yes, thank you very much. The validation work is very important. I really enjoyed your overview in this talk. But each validation experiment uses a single molecular structure as a reference. So how do we know that all structures expand the same amount, such as centrioles versus potassium channels or microtubules, or maybe different cell types in different tissues? So it's both a practical question, but also sort of a philosophical question, like, does it really matter for most work because we're not really comparing different structures? We might only be looking at what's going on in each structure at a time. Thank you.
Yeah, this is something that I think about too. In some of these work, they use 2. So, for example, I didn't show it, but in the one they did in my lab, they also stain the cytoskeleton, and also in the one in the ultra expansion microscopy protocol, they also stain. They use again the clathrin pit to observe another reference structure. But yeah, that's a very good question, and that's why at the end, my point is that there is not a universal method, because every work only look usually at a few structures at best, because you cannot examine everything in your sample, of course. But that's a very good question. I do not have an answer for that, but it's a very good question. It's something to think about.
All right, and you have a question in the chat. Have you seen a difference in the loss of signal after expansion between the different secondary antibodies, and which one do you use?
Yes. Other than the one that don't work, like for example, we know that some don't work at all. For example, the Alexa 647 doesn't work for expansion. All of the secondary antibodies I work with, I found that the Alexa 488 is the best for expansion, I think, at least for pre-expansion staining. Unfortunately, since I'm using GFP, I cannot use it. but yeah, and then I tried others. For example, the Acto647N for me has quite good retention. Also, the Acto594 is another antibodies that I'm using, and I also tried the 532, but I think the retention is a little less. I didn't show it here, but something that I started doing some time ago is to try to restain the secondary antibodies after the expansion. And usually this, I don't have the image in this presentation, but the signal to noise ratio gets a lot better, assuming your primary antibodies were not digested by the digestion buffer. Like if the primary survives, then the secondary staining can be a solution.
Okay, perfect. And one more question from Julie. She asked, what kind of mounting medium do you use for said acquisition on your gel?
Well, the gel is just enclosed between two cover slips, and then it's sealed with the component polymer, but like it's just to seal the gel to prevent the shrinking due to evaporation. But between the cover slip and the gel itself, there is nothing, like you imaging the gel on the cover slip. So that's what we do. And for the let's say not final fully expanded gel, we just do the imaging on the cover slip. For example, for expansion, since afterwards we want to expand it, we just image on the cover slip mounted in a metal ring with PBS on top.
That's pretty much the end of Chantal's talk. Yeah, so as I was saying, if anyone has any questions, we are happy to relay them to Chantal. Natalie actually asked her at the end of the recording, and she's happy for people to email her, or we can get all the questions together. That's pretty much what I was planning to do today, to get a bit of an interest of how many people would like to join this group. If anyone would like to volunteer as a speaker, that would be also much appreciated. And otherwise, our next meeting will be in 2 months, but there will be a meeting hosted by Canada in between, which might not be a very good time for us. Colleagues raising his hands.
Hey, Gabby.
Okay.
I just wanted to say, I think it's going to be, obviously we've got a lot of people that are interested in doing it, but probably haven't dipped their toes in the ocean yet. What we'll try and do in the meantime ahead of the next meeting is advertise it a bit more widely across Australia and Asia Pacific. And as you say, try and get a couple of volunteers who have experience to actually come and present, because I think that ability to interact and ask questions more directly, I think is going to be key to making this a useful group. But yeah, we'll try from the LMA sides to help you guys out.
Yeah, that would be great. So far, the help has come from RMS because it was sort of organized by Canada, so they were sort of organizing the website and things like that. But Pam did help me advertising with the mailing list. But yes, it would be great. The more people we get interested in sharing experiences, the better. And eventually we'll have enough information and people will be able to establish the technique. And maybe if Elena wants to give a presentation, that would also be helpful, because she seems to be the one that has experience in this technique as well.
Yes, Gabriela, I'll be happy to present because I have experience in different techniques, and I can also do like an overview of pros and cons, and if you're interested. Yeah, if you have, if you guys have any questions, so I'll
Yeah,
happy to answer.
That's great. Yeah, it's sort of, there's a lot of interest, but there's not a lot of people doing expansion in Australia.
Yeah, I know, but I just wanted to say to everyone, it's not scary to do expansion. It's like it seems complicated, but once you follow the protocol, it works. Because, for example, I work with Drosophila tissues, and I use protocols for cells on my tissue, and it works, like ultra expansion and fan expansion, and it works. You just need to follow the protocol, do exact the correct thing, maybe some some adjustments, but usually it works, and it's not as complicated as it sounds.
Yeah, that would be great. I think I'll contact you directly and try to organize.
Yeah,
maybe
sure.
The times, I'm thinking about the times, and maybe not great for you. Maybe we can find a time.
No, maybe you can move it like 2 hours forward. So it won't be 5 o'clock in the morning, it will be 7, so it's more convenient for me.
Yeah, no, sure. Yeah, that would be great. Well, thank you everyone for joining. And you've seen the document, the Google Doc. So if we haven't been able to answer questions, we can relay some of them to some of the Canadian part. And there's a question from Luis about the meeting for January 4th was like a tentative date. It was just more like 2 months from now. But I think it's probably more realistic to be mid of January because most Australians take a lot of January off. Yeah, we said yes. Yeah. And there's a question for you, Alina, if you have any experience with post-expansion labeling, like fluorescence in situ hybridization. That's from Pranali, like fish.
So some of the techniques actually, for example, pan expansion, ultra expansion, it's post-expansion labeling. But I used it for proteins. I didn't do in situ, but I know that some people do. It's supposed to, the nucleic acid is retained pretty well. I listened to the lecture now, and but it is a slight shift between the nucleic acid and the protein. From my experience, I used the Hoch labeling of the nucleus, and I saw that it slightly moved from the proteins that are surrounding it. So, but it's supposed to work. I think it's supposed to work. So for if DNA is retained, I guess RNA.
People would be really happy to hear from you. That's what they're saying in the chat.
Thank you.
I think if no one else has a question, I guess we'll close the meeting, and I hope to see you in 2 months' time. Okay.
Thanks, Gabriela and everyone.
Thank you.
Thanks, Gabby.